I would like to kick off this webinar, which is based for work package 7 actually, and it will show a couple of key results of the work package in the ConcePTION project, and then we're going to take a deep dive in two methodologies that will be presented by Rosa Gini and Judit Riera. Okay. So just for housekeeping, please, because we will be many, keep your microphone muted and post your questions in the chat. I do think you will be able to unmute when you want to say something, but we want to avoid that people start talking at the same time. So our speakers of today, my name is Miriam Sturkenboom. I'm a professor at the University Medical Center in Utrecht, and I will doing the introduction. I'm the project coordinator of IMI ConcePTION. And then my colleagues, Dr. Rosa Gini from Ars Toscana and Judit Riera Arnau will present the in-depth methodologies. Okay. So on behalf of the IMI ConcePTION Consortium, some background. What we are currently facing is that more than 60 years after thalidomide, which really started the regulatory system in many cases for safety monitoring, we are still facing the fact that we have a big problem to solve in the pregnancy area. So in Europe alone, 5 million women become pregnant in each year. Up to 90% take medication at some stage during their pregnancy or when they are breastfeeding. But because of the lack of information that is currently out there, or lack of evidence, solid evidence in the package insert, 1 in 3 chooses to discontinue treatment, which may have potentially serious consequences to health. And 25% of pregnant people actually experience anxiety due to lack of information about their medicines. And since they take a decision for 2, it creates anxiety. So in 2019, the IMI ConcePTION project was started. It was funded by the European Commission's Innovative Medicines Initiative, and it's a consortium of more than 80 public and private sector organizations that are drawn from academia, regulatory agencies, and global industry. We came together united because we have a common belief, namely that we have an important societal obligation to radically and rapidly reduce uncertainty about the effects of medications that are used during pregnancy and breastfeeding. And in the project, we aim to achieve this by establishing a trusted ecosystem that generates and disseminates reliable evidence-based information regarding the effects of medicines. So how do we generate evidence on medicine safety in pregnancy from electronic healthcare record, which is the focus of today's webinar? This is done through the ConcePTION pipeline. Within the project, there are various streams of information or evidence generation, and the electronic health records are one of them. So here you see the project flow as we designed it to build a learning healthcare system. On the left-hand side, you see that ConcePTION dealt with various types of data. Amongst, health data is one of them, but there was also animal and model data, pregnancy reports, milk and blood samples, women reporting in prospective manners. But today the focus will be on real-world data from reuse of existing healthcare data. What is needed to build such an ecosystem that we wanted to build is the various components that you see from left to right, such as a data catalog that describes the metadata of available data. Data access needs to be arranged, data harmonization, quality management, before you can actually do the evidence generation on signal detection and rapid assessment. And then, of course, at the end, we do not only want to generate evidence, we also want to disseminate it to the healthcare professionals and pregnant people, and that is conducted through a knowledge bank that was also established. So today's focus is going to be limited to real-world data and on certain aspects that are needed to actually do these studies. So a very quick overview of Work Package 7 output was that the FAIR Meta data catalog was created based on the Molgenis platform, an open source tool that is made by the University Medical Center in Groningen. And they included in their data catalog the ability to describe also routine use healthcare data sources. So that's organized through organizations, data sources, data banks, network studies, and data models. And this catalog was the basis also for the Minerva project funded by the EMA that led to the new HMA-EMA catalog. The ConcePTION EHR data approach is built on a distributed model where people use a common protocol, a common data model, that is the ConcePTION common data model, and common analytics. So the data stay local to comply with GDPR. No data going to the analysis goes to the data. So, and we use local knowledge about the data, which is essential in the harmonization step. In the beginning of the project, we created the ConcePTION common data model. This was also published, a very similar structure as other common data models, such as the Sentinel and the OMOP common data model, but made, including very much the pregnancy related needs, actually. And this common data model has been utilized throughout the project. The data transformation pipeline for the real-world data takes a series of steps. It starts on the left-hand side in the green. You see the data sets. D1 is the native data model that is locally available at the data holder. And they use local expertise to create and design to transform the data into the common data model, the ConcePTION common data model, which is D2. And that they do through an ETL script, first a design and then a script, and then subsequently data scientists, epidemiologists, and statisticians create scripts that do the steps T2, T3, and T4, which are semantic harmonization, application of the study design, statistical analysis, and then at the end, the results are being shipped to a workspace where they are being pulled to put in a report. So that's the transformation pipeline that is being utilized. Work Package 7 also created a data quality script. So when the data is transformed from the D1, the local data, into the ConcePTION common data model, we want to ensure that this is done correctly. So there's a whole pipeline of checks that is being done from level 1 to 3. With more than 600 indicators that can be utilized to inspect whether the data is of good quality and fit for purpose for the study. These scripts are utilized heavily by different research networks beyond ConcePTION and have already shown their need and effectiveness. We created an open source toolbox for this real-world data transformation pipeline, which is utilized. There is a wiki for ConcePTION where you can find access and links to all of the tools that are available, including the GitHub, where we have functionalities. And it is reachable also from the imi-conception.eu website. So if people want to find it, you can link to that. This pipeline for transformation of real-world data has been utilized in different post-authorization safety studies and EMA framework contracts, and has shown its effectiveness also in the area of the COVID, for example. It was utilized both in vaccine studies, but also in medication studies in pregnancy, where we assessed for EMA that medication utilization and also what was done in hospitalized and non-hospitalized women, and this could be compared and actually pooled even with US and Canadian data. So this is very quickly an overview of core Work Package 7 output in the ConcePTION project. I would like to now give the word to my colleagues to go in more detail in specific steps of the pipeline.
Good afternoon, everybody, and thank you for the opportunity to to illustrate this important work that was conducted in the ConcePTION project. And thanks to Miriam for introducing it. I would just like first to clarify that the ConcePTION project has really extended further its own consortium already. And this is the list of, it's the pregnancy algorithm team that really involved many institutions, even outside of the ConcePTION consortium, because of the interest of the activity that Miriam explained. And many projects have already used the preliminary versions of this algorithm, and their institutions are included in this work. And this is, well, of course, the disclosure of the project. So this is the background of the ConcePTION project that Miriam described already, and the ConcePTION pregnancy algorithm. So as you know, ConcePTION is an infrastructural problem. So project. So not only we were going to extract pregnancies from the data sources that were participated in the project, but we really designed meta algorithm trying to highlight which are the conceptual aspects that you need to tune to every diverse data source in order to first identify in the most comprehensive and timely manner all the pregnancies that are included in a specific instance of the data source, regardless of how the data source is composed or how the pregnancy is ending. And to assign to each pregnancy in the most reliable manner a start date, which for us is the date of the last menstrual period, the end date, and the type of end of the pregnancy. So which is not really the outcome, but we have a classification that we will illustrate in a moment. And this was done taking into account the diversity of the data sources and creating a kind of transparent framework in which any possible data source can be fit. And this is a bit complicated because the part of the slide is hidden, but I think the title of this is the documentation of the pregnancy algorithm. I leave here, and the slides will be shared, some publicly accessible links where the pregnancy algorithm is illustrated already, together with its resources. And there is a manuscript that is in preparation. So I want to dedicate most of the presentation to the overall design of this meta algorithm, because of course it has been a long work and we have identified many, let's say, nuances that didn't meet the eye. And we believe that we have captured them in a manner that, of course, everything can be improved, but we think this is really interesting, and I want to take this opportunity to enter into a bit of a detail. So what is the frame? So the first step of our algorithm is retrieving from, so we have a instance of the data source that is converted to the ConcePTION common data model for practical purposes, and we retrieve a set of dates. Okay, what does it mean? How do we do that? We retrieve all the dates of all the records that may imply that the person about whom that record is recorded is pregnant on that day. And then we associate, so we somehow elaborate on the semantic, on the implication of each record. So some records, which are our favorite, of course, imply that the pregnancy has ended on the record date, and also carry into the record itself information on the gestational age at that date. So they give us the possibility to identify the end and the start. So the circle is the start of the pregnancy for us. And they are green. So the quality of those records is perfect. Some records do imply that on the date when they are observed, the pregnancy has ended on that day. But they do not carry explicitly information on gestational age at that date. We label those records as yellow, and we record that we know what the end date of that pregnancy is, but we cannot put a circle, you see? So we don't know. So we create provisionally a length of pregnancy with some rules that I will show in a second. Some records imply that on the record date, the pregnancy is ongoing. So it's not ended or may still be ongoing, but they do carry information on gestational age at that date. So we call them blue. And so in that case, we can assign a circle to our pregnancy, but we need to guess somehow on the end because we actually don't know. It may be also in the future. For sure, it may be in the future of the record date. And then many records do not carry any specific information, neither on start or on end date of pregnancy, but they carry the information only that on that date, the person is pregnant. Okay, and we call them red. And we create and start on an end according to some rules. As I mentioned earlier, we have 40 code list of diagnostic codes, and this is the names of our the lists of diagnostic codes. And according to the diagnostic codes, which define some specific events, we know whether the type of end is recorded, that is live birth or not, and it's unknown, or it is still stillbirth. All of them imply that the pregnancy is ongoing at the record date, but some of them also imply that the pregnancy has ended at the record date. And we have a list of 40 code lists with these implications. Then we have lists of procedural codes, like, I don't know, amniocentesis. And then we have lists of item sets. So many data sources have birth registries, and in that case, information is not coded. There are question and answers, and this also we also retrieve. So first step, retrieval and assignment of start of end, let's say, based on the logic of the specific record. Second step, exclusion criteria. We eliminate the records whose person is not recorded in the list of persons included in the data source that may be available somewhere else. If the person does not have a birth date or is not in fertile age at the start of pregnancy, and if the record date is not included in the period when the person itself is observed in the data source. We do not exclude records assigned to person of gender that is not female. Now, we have all of our records. Well, at this point, we need to create the pregnancies. And what does that mean? It means that we group all the records of the same persons that are around of the same date and that probably or may belong to the same pregnancy. And this is where the devil is really in the detail. This is a very complex process, and it really profits from the expertise of the local data expert in order to make decisions. And the decisions are a bit more complex than that, but not, I mean, if you go in the deliverable, they're really explained in detail. But let's say that the majority of the variability are captured by the two parameters that I'm explaining now. So this is the period after or before a pregnancy in which pregnancies are implausible, and we set them at 28 days after discussion with our expert. It means that if we observe a pregnancy for example, within 28 days after birth registry record, we believe that it is a follow-up visit. Okay? And then gap allowed, this is specific of the data source, is the maximum time that you can allow between two records when the person is pregnant before saying, there are definitely different pregnancies. And this really changes according to which data you have. Because if, for example, if you expect that the end of pregnancy does not create a record, for example, in a primary care data source, you can expect that there are long gaps between pregnancies. If you have a birth registry, for example, and you see two records many records of pregnancy along a long period where it's implausible that it's a single pregnancy, it's most probably many miscarriages. So this type of decisions, they are decided with the expertise of the local expert. At this point, we do the first attempt to avoid overlapping pregnancy. And there are some basic rules that we have according to whether the record is just put the yellow and green, but we also try to reconcile. We have rules for the blue, for the red, etc. And so we separate pregnancy at the using information from the record. We create a predictive model. So in some data sources where we have green records or blue records, we learn the beginning of the red pregnancy and of the yellow, so of the red records and of the yellow records based on the green and the blue. And then we predict it. So let's say we have our previous examples. We use the green and the blue to predict this, to learn about the red and the yellow, that this is the start. And then we use this information to predict it for the red. And then accordingly, we adjust the end so that all the records contributing to the same pregnancy are reconciled. And at this point, we may have created further overlap, and we have rules to avoid overlap. So the list of pregnancies from the pregnancy algorithm output do not overlap ever. And what is the output? First, the final data set of pregnancies with our main variables, start date, end date, type of pregnancy end. And then we include many variables that tell you how is it that we came to the decision of assigning that start date and that end date and that type of end. So that not only, so you have understood now that we have the color code, the red, the blue, the yellow, and the green, but these are just the main quality variables. We have many. So that somehow in your study design, you can also decide that some pregnancies whose decisions were particularly delicate, you discard or you verify, because the output comes with a sample of pregnancies that the local expert can verify manually. And also there is a functionality to include, let's say on purpose for the local expert, the more information on how the pregnancy was created of any list of pregnancies, so that the local expert can confirm or disconfirm the choices that the algorithm made. Moreover, we have a data set where if the data source does have a link between mother and child, the pregnancy algorithm further enhances it with a link between pregnancy and child, because of course the same woman may have multiple pregnancies, and this is assigned by the algorithm. And then we have a descriptive markdown report that gives the information about, a descriptive information about the cohort. And this is the classification of type of end. And we don't have only live birth, stillbirth, spontaneous abortion, termination, and ectopic or molar pregnancy. We have many others. We have pregnancies where we don't know anything about the end. Could be a live birth, could be a termination. We just don't know. We have pregnancies where we know that the pregnancy ended early. So somehow the outcome, the type of end was unfavorable somehow, but we don't know whether it was a termination, a miscarriage, or maybe a stillbirth can also be included in this type. Then we have birth unspecified. It means that we don't know if it is a live birth or a stillbirth. And then we have unspecified, which means that we do know that the pregnancy ended on that day, but we don't know anything else about the end of pregnancy. Then there is the case when the pregnancy is still ongoing at the moment when the pregnancy, when the data source was extracted, when the instance was extracted. And this is a very valuable set of pregnancies that can be used for studies that are somehow done in an emergency, where you want to know what's going on with pregnancies that are, for instance, whether they are taking a new drug or a new vaccine. And then lots to follow up. It means that the person that is pregnant left the data source before the pregnancy had a chance to end. And two of those outcomes are not implemented in the version of the algorithm that is included in the manuscript, but they are in the new version. Now, this was the methods, and the tool is available online, can be adapted to any data source. We can support that. Now I'm going to explain briefly how it was tested in 10 data sources. And this test actually somehow informed the creation of the algorithm. These are the, well, geographical diversity across data sources that participated. In order to handle diversity, which was the really extreme, we utilized the diverse framework that was finalized last year by a ISPI group. You have 9 dimensions to characterize a data source. The type of organization that makes the data accessible to research, the institution that creates the data, what creates the data itself, what prompts the data into existence, what are the rules to include the population, what is the content of the data, what is the data dictionary, including the data model, what is the time span when the data source is available, what are healthcare system and cultural characteristics that are pertinent, and what is the data quality of the original data for its original purpose. And we have mapped all the 10 data sources to the diverse framework, and this is also publicly available already. For example, in the case of the Norwegian data, we further split it into 3 data banks, we call them, because they have different origin and different prompts. So in this case, it was the birth registry, of course, the hospitalization record, and the reimbursement for primary care services. But all the 10 data sources were split differently. The characteristic of healthcare and culture have an enormous impact. For example, in this case, it is mandatory to report information about pregnancies with gestational age 12 weeks and beyond, and this is not true in other data sources. For example, I will not enter in further detail, but of course you know that in the Netherlands, there are many women that do not deliver in hospital, and of course this needs to be taken into account and has an impact on the results of the algorithm. Overall, the diversity, if we just split all the data sources into their data banks, you can see that the combination, there are no 2 data sources with the same combination of data banks. And yeah, this is why we had to work so much to capture all this. Now, results. So this I really don't see my slide, but let's say that this is the type of numbers that we obtain at the end. We just studied pregnancies from 2015 to 2019. So we took a span of 5 years before COVID. So we were, let's say, on the safe side from what regard that. And sorry. And this that you see here, I will just give you the color code of the pregnancy, and you see that the variability is enormous, both in terms of inclusion, and of course it is projected into the type of ends, where, well, only one data source had ongoing pregnancy because it was capped early, but you see that some pregnancy have unfavorable outcomes clarified. Many have large span of yellow data sources, yellow pregnancies, and some of them are have a large share of unknown type of ends, and accordingly to, or termination, and accordingly to the type of data sources, unknown type of ends may consider to be most probably spontaneous abortions that happen early because other types of end would be captured. This is the results from the prediction model. The root mean square deviation could be roughly interpreted as the error in the estimation of the start of pregnancy. If the records that predict the start are yellow, the error is smaller. If they are red, the error is larger. So if they are yellow, the error is, let's say, less, in sometimes less than 2 weeks, which is pretty reliable. In the case of red, it's a month in the best case scenario. And this, of course, needs to be taken into account in interpreting the results of a study. A very important fact is that we are able to identify pregnancies very early, as early as 47, 48 days, which means that, of course, we will be able to capture early miscarriages, which is not possible in other data sources because, well, there are extreme cases where you really capture the pregnancy only at the end. But yeah, so these numbers are really, really very important according to whether you want to study miscarriages as an outcome or not. On an example of application that we believe is important, in November 2021, almost 2 years after the COVID pandemic started, the ConcePTION project delivered to EMA a first report on medicine utilization for COVID in pregnancy, and the 2 data sources that could apply the ConcePTION pregnancy algorithm were able to identify hundreds of cases of pregnancies affected by COVID during the first 2 trimesters and to report on treatment. So, conclusion. We have created a meta algorithm that handles in a transparent manner data source diversity. We have tested it in 10 diverse European data sources. The distribution of quality and type of end is influenced heavily by data diversity, and the decision on which pregnancies are included in a study should be based on the data source characteristics and on the research question, and with the aim to minimize bias in the results. And I would stop here.
Thank you everyone for joining. And even though the first half of the webinar I see has been really active and intense, I hope that you are ready to stay ahead a bit more because I will speak about the tasks of working package 7, specifically the task about teratogenic drug prioritization, that is the 7.9. Let me remark that I will be the speaker today, but this work is just being possible thanks to a bigger team, and would like to thank especially to Emma Alzina. And if there are any questions, I may answer them at the end because we will have some minutes for a Q&A. And this is the content I will go through, starting with a bit of background on the fundaments of this idea we had, and later explaining the results we got. I have divided the results into the two main sections of this project. First, a descriptive study, and after the algorithm development. You will understand this a bit more in a few minutes. So starting with the background, well, as you may know and others have already remarked, the proportion of pregnant women who take medicines, as well as the number of medicines that these women take, has increased greatly in the last years. And exact data is lacking, but some estimates indicate that around 64 to 90% in 2015 of pregnant women take medicines during pregnancy. Then some medicines are currently thoroughly known to induce long-term teratogenic or neurodevelopmental effects on human fetuses, and have often also included teratogenicity studies in rats or rabbits. So their effect were already known by embryo-fetal developmental toxicity studies. I will call them EFDT. In regards of safety concerns, 91% of the FDA-approved medicines before 2010 lacked data about safety for pregnancy and lactation intake. So what we can learn from this past evidence is that as some of these drugs have already shown teratogenic effects in the EDFT studies, can we learn something from it? Because, for example, valproate, a medicine that was licensed to treat epilepsy and bipolar disorder many years ago, already showed teratogenicity in animal studies around the 80s. Therefore, we think that we may have learned something from these studies and used this for the human protection, because as you know, currently valproate is an issue of interest, and many studies are targeting it. Okay, here I would like to present this specific scheme from a specific project from Thenissen and colleagues, because the Dutch National Institute of Public Health and Environment a few years ago created a database comprising rat and rabbit EFDT studies and their outcomes in regards of teratogenicity. This study using the animals was performed by Dr. Thenissen, and they included compounds which applied to a registry or were known teratogenic. Then they exposed rats or rabbits to them and took a set of items in consideration to establish a kind of a set of categories depending on the concerns they indicated about the teratogenic effects they may have. Here, as you can see, upper left in the slide, around 200 medications applied for registry, and around 20 were already known teratogenic human pharmaceuticals. They analyzed them on rats and rabbits. Some of them were used in one of these species, and the others were used in both of these species. They monitored a series of items like the MEFL, for example, and the AUC, the area under cover, and the NOAEL, the non-observed adverse events level. And from this information, they established 4 categories. First, the red one that indicates that should be a drug of increased concern. Then the yellow and orange, which are of intermediate and lower concern, and then the green drugs, which are of minor concern. Here in the right side of the slide, you can see the specific items that were considered. You can see, first of all, the positivity for malformations or any embryo-fetal death. They included mainly major malformations, but also some minors. Then the exposure margin calculated by the area under cover, and also the NOAEL level of conversion to the human species. Then the compounds showing an embryo-fetal death or major malformations with an exposure margin of less than 10-fold exposure are considered to pose also an increased concern. Then this threshold changes when it's 10 to 25-fold or more than 25-fold. So the results from applying this algorithm, you can see it to this snippet of table. You will note that not all the drugs have been included for the sake of space in the slide. But in this table, you can see each of the items I mentioned in the column titles, and then each compound that was tested in the rows. They have been sorted, showing the ranking of concern that they showed. And here in the third and fourth column, you can see the species where they were tested, the outcome of any malformation or death that they got, a 0 if nothing was seen, and 1 if that malformation or death was seen. And in the latest columns, you can see the area under cover and the ratio of conversion of the level of dose from rats to humans. And from all this information, they have established this ranking of concern. Perfect. Just in summary, because the previous names were a bit small, some examples of the drugs that they detected as relevant included anti-neoplastic agents, also anti-epileptics, retinoids as well, and other antibiotics or hormonal agents. Okay, so with all this in mind, which were our objectives? So we believe that taking advantage of the animal studies and real-world evidence data from humans could help prioritize the performance of safety studies for potentially teratogenic medicines. The first question we ask ourselves is, what is the prevalence of prescriptions of medicines known to be associated with any developmental toxicity in women of childbearing age or pregnant women? We divided this research question into 2 steps. First, we performed a descriptive study to discover and determine the prevalence of the medication used in pregnant women and women of childbearing age. And as a second step, from taking the information from this previous work from Teunissen and also from the results of the descriptive study, we aimed to develop an algorithm to rank these medications for humans and integrating the information of all the previous. So going now to the methods. First, the methods for the descriptive study are that we performed a descriptive and retrospective cohort study and included the databases Ferrara, Fisabio, and CDAP. They are from Spain and Italy. The criteria of inclusion were women of childbearing potential. And to identify pregnant women, we used the algorithm that Rosa just explained and selected the most certain pregnancies, let's say, no? The yellow and green ones. As outcomes, we analyzed the yearly prevalences of drug use in women of childbearing potential with 1 or more prescriptions or dispensings. And for pregnant women, we assessed the prevalence per trimester of pregnancy. Okay, here you are the calculations and the definitions of the trimesters that we used. Okay, and in regards of the methods for the second part to develop the algorithm, we just integrated the knowledge from the preclinical study and our prevalence study, and then we established a set of clinical criteria and considered a series of winning factors. These decisions were a mixture of clinically driven and also data-driven. Okay. So now let's see the results for each of the steps in the study. First, in regards of the descriptive study for CDAP, we keep in mind that in these graphs, we are only seeing the top 10 medicines we found more prevalent. And here in the case of CDAP from Catalonia, we see that ibuprofen is clearly the winner here, and that the percent of usage has been really steady over years. We see a bit of a drop in the last year in 2020, but it might be because in our data this was not fully complete, but it seems that it remains steady over time. And then the second here, it's the aspirin, acetyl salicylic acid, which shows really discrete increase. But you can see that the prevalences are really, really low, around 1%. So it's difficult to appreciate. For Fisabio, we find comparable and consistent information than in CDAP. This makes sense as both are in Spain. And we see that ibuprofen has steeply increased, especially in 2019, reaching the 20% prevalence. And aspirin has also had a really discrete increase. And the second one we see here, the JO2, bottom left, it's the fluconazole, which is an antifungal agent. And finally, it was impressive for Ferrara. All the prevalences we found in women of childbearing age, all of them were really under the 1%. Just to comment something, if we zoom in the methotrexate and codeine and combined with ibuprofen, we see a bit of an increase, but you can see that this is really insignificant, taking into account the overall percentage. Now, going to the usage in pregnant women per trimester. You can see the red bars refer to the first trimester of pregnancy, green bars are for the second, and blue bars for the third trimester of pregnancy. Here, the first one is ibuprofen as well, and the second one is aspirin. And just to have a few numbers here, the overall prevalence in the case of ibuprofen, it's more or less 1.7%, and the highest usage happened during the first trimester. In the case of acetyl salicylic acid, it was in the second trimester, and for fluconazole, which is the third in the plot, we can see that the overall prevalence is around 0.1, and the highest usage was in the first trimester as well. And for all other medications, prevalence was really less than 0.1%. Also, this is calculated over a total of 500,000 pregnant women. Okay, then for Fisabio, the results are similar, but the pattern across trimesters changes a bit. For ibuprofen, we see a similar prevalence than in CDAP and for aspirin as well. But for ibuprofen, here we found that also the usage was highest in the first trimester, and for aspirin, the second trimester. And the third, it's also fluconazole, but was highly used in the first trimester. And then for Ferrara, it's really difficult to notice something. They had information about 370,000 pregnant women. Valproate and carbamazepine here are the first ones, so they could seem to be the highest, but you can see that the difference is not that much. And one point we would like to make here is that in this case, really small changes may change a lot, very differently, the order of these drugs in the ranking and in this plot. So yeah, if some drug that is in the 11th position could be easily in the 3rd position with small changes happening. And in this case, just to remark a few, I found that for ramipril, cotrimoxazole, and carbamazepine, the highest usage was in the first trimester.
Judith, may I
Yeah,
add something?
Sure.
It's
Absolutely.
important to keep in mind, comparing the Spanish and Italian data sources, that the code lists for each data source are tailored to the DAP. So Ferrara does not, they did not include ibuprofen in their code lists. So it's not even that the prevalence of ibuprofen was so low, it's that it wasn't part of the research question for that particular DAP.
Mm-hmm.
But if I may comment, the reason for that is that data source only captures reimbursed
Exactly.
drugs. So
exactly.
it's
Yeah.
the reason why it was not in the code list is that it is not in the data
not
source.
captured.
Exactly. Yeah. So when we're thinking about the Spanish versus the Italian and why these graphs are so different and the prevalences are so low, in Ferrara, it's because the Italian code list was radically different and the Italian reimbursement system is radically different. So thank you.
As you thank you, guys. Thank you, girls. Because as you remarked, we also should keep into account the prompts that cause a record in each of these databases, because this may also make a difference. So it's really important to know which type of database we're working with, no? Prescription, dispensing, and especially with ibuprofen, which was the most remarkable one. It's OTC and on demand in some countries, and this may vary across healthcare systems. So yeah, just to compare a bit more here between the three tabs, you can see that the Spanish databases seem consistent more or less between them. This tells us that there's an important role of the social and health context, and also all the confidence intervals are really small. So yeah, we can say that we trust these prevalences, keeping in mind the points that Emma and Rosa commented. Also, in regards of the indications of the top drugs, which is the sense, which is the reason why we find that ibuprofen and aspirin are the highest ones, especially in Spain. So one explanation might be the indication, because there's a kind of belief that there are really a few therapeutical options for pain during pregnancy, and maybe during the first trimester. Women do not even know that they were pregnant, so they are still using some treatments. And as ibuprofen is on demand, yeah, this can make a huge variation. And also, at least in Spain, there are different dosages of ibuprofen that are captured differently. So, for example, a dose of 600 milligrams needs to be prescribed, while a dose of 400 milligrams doesn't need to be prescribed. This can be also a difference if we could look at the dosage. And then aspirin, it's frequently prescribed at lower doses to prevent preeclampsia during pregnancy. So in this case, depending on the dosage, this could also make a therapeutical sense in a way. And finally, yeah, just to remark that in Ferrara, low prevalences may cause that small differences position drugs very differently. Okay, now jumping to the results of the algorithm development. Oh, I'm sorry. Now, perfect. So we have built this pyramid. Oh, sorry. We have built this funnel, including the different criteria that we included in the algorithm. All these criteria are clinically driven and also data driven, because from the results that we got from the prevalence studies, we established a set of definitions and thresholds for each of these criteria to try to make it more relevant. The first point was the percentage of pregnancies being exposed, and this is the bigger one because we gave it a bit more weight than the others. And the reason was that the higher the prevalence, the greater the risk of fetal exposure. In second place, the percentage of pregnancies with more than one exposure, because this may relate to multiple prescriptions suggesting a higher accumulative dose or longer exposure, which may came out with severer outcomes for the fetus. Then percentage of pregnancies exposed during the first trimester, because this is especially concerning trimester, as this is the period when the major organ development occurs. Then the percentage of exposed women of childbearing age, because again, a higher prevalence in this collective indicates a potential higher exposure in pregnant women. And finally, the positive trend of usage of the medication, because a rising trend in the use of a drug over time may suggest an increasing exposure in the population, and also that social changes might be happening there. Okay. So for each of these criteria, we established a set of cutoffs that varied slightly depending on the weight that we decided to impute to that item. In the table of the right side, you can see that, for example, in a percentage of exposed women, we used cutoffs going from the 0.1%, but then we lowered it to 0.01% because of the really low prevalences that we found. Here in the blue-headed table at the left, you can see the priority and the score that we assigned to each of these ranks. And so the 0 points were for the first cutoff, then 1 point for the second cutoff, and so on. So the more points, the more concerning the drug would be. And okay, and finally here in the green-headed table, you find the preclinical multiplier. This is the way that we incorporated the preclinical information to our algorithm. So if the classification from the preclinical animal study was green, we multiplied the score of the clinical side of it per 0.5. And then if it had a red, so a more severe or death outcomes in the animals, or a NOEL rate conversion more nearer to the human levels, we multiplied per 2 the score. Okay, and here you can see also what we got. So in the column names, you can find each of the criteria that I just explained for the algorithm, and then in the 3 final columns, you can find the scores that that each of the drugs got. In the 3rd column, starting for the end, we have the clinical score. Then we have the preclinical multiplier, so the color code that I just explained, and then the final score that came multiplying these 2. So if we look for each of the databases that were included, we can easily see that ibuprofen, aspirin, and fluconazole are between the highest scores, so the more concerning, I would say, especially in Spain. This relates to the high prevalences that we have seen previously. And in the case of Ferrara, where the prevalences were more decimal, I would say, the final scores might be lower, but they remark the anti-seizure medications. Also to interpret, we also can comment that lower scores came out to be for liraglutide, for example, and other antivirals, which have 3 points, 2 points here. Also, what we may say is that the positive significant use trend has not contributed very much to the final weight to the score. But all the outcomes in the pregnant population, so the exposure during first trimester, the more than one prescription, and the general exposure in pregnant women had a lot to do, because you can see that the highest scores here correspond with the highest final scores. And also the preclinical multipliers were of 2-fold. So how do we interpret all this? First, we need to remember the intention of this algorithm. This algorithm is just proposed as a way of synthesizing information coming from different sources, so the preclinical and the clinical settings, and translate it into a quantifiable value to ease the comparison within a DAP and across medications and between the apps. The criteria that we were discussing to have for the final score, data-driven absolutely, is that medicines with a low than 6 score are of low priority to perform pharmacoepi safety studies, 6 to 10 middle, and over 10 high or urgent priority to target. But the scores finally need clinical interpretation to make sure that they are relevant for the society and the setting we aim to apply that. So this is just a question to the audience. In short, we will have the Q&A session, but we would kindly ask feedback about the criteria, the weighting that we decided, the calculations, because we feel that your varied expertise can help a lot by nourishing this algorithm, improving it, and having it ready for the final report that we hope to deliver soon. Just to comment some strengths and limitations of this work, before jumping to them, I would like to comment about this paper that recently was published by a team from the US, and it's pretty similar to our algorithm, but it's a protocol that maybe some of you already know that publication, but it's about a protocol to also establish an algorithm, but they focus on risk mitigation strategies and to prioritize them for a set of medicines. So it has more regulatory perspective would say, and they also only rely on claims data and ad hoc analysis. It's really interesting to see that it's a field of interest abroad. This shows that we have common worries, but however, you may appreciate that our algorithm has a slightly different scope and input and aims to consider also the preclinical information and together with the descriptive information about using real-world evidence. Yeah, so the algorithm we created may help researchers to prioritize the studies, but not necessarily regulatory actions. We account for the ratio of conversion across the species, which is also concerning when a clinical trial or a medicine like this is evaluated. We include real evidence of use together with in the algorithm together with preclinical data, and the only aim is to synthesize diverse information into a quantitative value that it's straightforward to interpret. But all the teratogenic drugs have not been included in the animals categorization, so we restricted the drugs included in the algorithm to those that were available in the preclinical one. Some of the drugs that were tested in the animals are not commercialized or are not available for humans, and we should take care and cautiously interpret this applied to other settings rather than Spain or Italy. So yeah, in summary, we see that ibuprofen and aspirin are amongst the first in use here, that in Ferrara, the overall use of medications is really low, but might be due to characteristics, intrinsic characteristics and prompts of the data, and the combination of information that the algorithm offers. So that's everything I wanted to explain. I would be glad to take any questions. Thank you.